Dolly the sheep's first cloned mammal in the world. Today we are going to discuss about 5 content from Dolly the sheep.
First, let me talk about the introduction of cloning. Cloning is the process of generating a genetically identical copy of a cell or an organism. But today, we are focusing on animal cloning. Animal cloning is the process of creating genetically identical individuals, mostly through somatic cell nuclear transfer, which is SCNT, which involve transferring nuclei from in vivo or in vitro derived somatic cells into recipient enucleated oocytes.
Hello everyone, so we continue about the lifetime of Dolly the sheep. On 5th July 1996, Dolly was born at Roslin Institute in Scotland, and she was the first mammal to have been successfully cloned from an adult cell. Dolly's name was a code name, which is 6LL3, but she took the name after a singer and actress at that time, which is Dolly Parton. So Dolly was publicly announced in February 1997, and supporters say that cloning technologies will have crucial advance in medicine which will bring goods to humankind. In January 2002, Dolly was found to have arthritis in her hind legs, a diagnosis that have raised questions about genetic abnormalities that may have been caused from cloning process. So after suffering from progressive lung disease, Dolly was put down on February 13, 2003 at the age of 6.
Next, I will explain what happened during the cloning process of the Dolly sheep. Before that, to make it easier for you to understand, I will give a name to the nucleus donor sheep as sheep 1, while the egg cell donor sheep as sheep 2. Remember that the sheep 1 has white face, while sheep 2 has a black face. Firstly, the mammary gland cell was taken from the sheep 1, which is the fin dorsate ewe. After that, an egg or ovum from the sheep 2, which is Scottish blackface ewe, was removed. Next, once the ovum had been enucleated, it was then fused with mammary cell from the sheep 1 by an electrical pulse, in effect emulating the process of a sperm entering the egg, but with both copies of the chromosomes coming from the same parent sheep, which is sheep 1. So the newly fertilized egg is genetically identical with the single parent. When fusion has taken place, chemical factors in the egg are released to stimulate cell division. Then, after the cell had multiplied sufficiently to form the making of an embryo, the new cell cluster was transplanted into the womb of a surrogate mother, which is the other female adult Scottish blackface ewe, which we name it as sheep 3 for a 21-week gestation period. Finally, a sheep called Dolly was born on the 5th of July 1996 by caesarean section and was found to be genetically identical to sheep 1, from whose other her DNA originated.
Hi and Assalamualaikum. My name is Siti Nabilah Huda Binti Muhammad Faizal, and today I will talk about the advancement of cloning method. Cloning of stem cell will provide treatment for a variety of disease. So what can cloned stem cell be used for? The stem cell can be induced to differentiate into different types of cell as needed. Next, the possibility of resurrecting an extinct animal. So, are there any extinct animal that has been cloned? Yes, a cloned French ibex was born on July 30 on 2003 in Spain, but died several minutes later due to physical defect in the lungs. Next, the possibility of selecting high-quality embryos before transfer. By using this system, we succeeded in selecting chromosomally normal clone embryos and could improve the cloning success rate after embryo transfer. So if this selection and epigenetic modification method could be combined, exploration of the mechanism involved in genomic reprogramming would be accelerated, and question about the difference between embryonic cells-derived clone and SCNT-derived clones might be solvable. Last but not least, nuclear transfer technique for analyzing germ cell epigenetic. Pre-meiotic germ cells can also be used for constructing diploid embryos by NT, and these can work as a powerful tool to determine the dynamic of epigenetic change during germ cell development. One important study in this category is the analysis of the genomic imprinting status of germ cells, especially primordial germ cells, which is also known as PGCS, and genocytes.
First, you need tube cardboard, small box, paper tape, some ropes, glue, and cotton ball from teddy bear. So you need to attach box and tube and apply the glue on it. After that, put cotton balls all over the box. Roll the rope around cotton ball to make its eye. And we are done, Dolly the sheep. That's all from us. Thank you for listening. Bye bye.